Swastiastu. Good morning, sir, and good morning, everyone. Today, I want to present about lipid metabolism. Before that, let me introduce myself. My name is Putumera Chandra Cahyani with license 1011. So, the first question about concept of lipid metabolism. Explain the definition of lipid metabolism and its role in providing energy for the body. Lipid metabolism refers to the biochemical processes involved in the synthesis and degradation of lipid in the body. These processes include the breakdown of fatty acids to produce energy and the formation of lipids for energy storage. Lipids serve as a major source of long-term energy because they yield a high amount of ATP when oxidized. Therefore, lipid metabolism plays an essential role in maintaining energy balance, especially during fasting or prolonged physical activity. The next question about fatty acid degradation or beta oxidation. Explain the main stage of beta oxidation of fatty acid in the cell, including the location of the process. Beta oxidation is the process of breaking down fatty acid into acetyl CoA units. This process occurs in the mitochondrial matrix. The main stage include activation of fatty acid in the cytoplasm, transport into the mitochondria via the carnitine shuttle, and sequential removal of two carbon units in the form of acetyl CoA. Each cycle produces NADH and FADH2, which are used in the electron transport chain to generate ATP. Next question, analysis of beta oxidation disorders. Based on the case, why can decreased fatty acid oxidation lead to fat accumulation in the body? Decreased fatty acid oxidation means that fatty acids are not efficiently broken down to produce energy. As a result, these fatty acids accumulate and are re-esterified into triacylglycerol, leading to fat storage in tissues such as the liver and adipose tissues. This condition contributes to obesity and fatty liver, as seen in the case. Next question about fatty acid synthesis. Explain the process of fatty acid synthesis, including location in the cell, main substrates, and important enzymes. Fatty acid synthesis occurs in the cytoplasm of cells, mainly in the liver and adipose tissues. The main substrate is acetyl CoA, which is converted into malonyl CoA by the enzyme acetyl-CoA carboxylase. The enzyme fatty acid synthesis then catalyzes the formation of long-chain fatty acids such as palmitate. This process requires ATP and NADPH as the energy and reducing power. The next question about difference between degradation and synthesis. Compare beta oxidation and fatty acid synthesis in terms of location, function, and energy requirement. Beta oxidation occurs in the mitochondria and functions to break down fatty acid to produce energy in the form of ATP. In contrast, fatty acid synthesis occurs in the cytoplasm and functions to store excess energy by forming fatty acids. Beta oxidation generates energy, while fatty acid synthesis requires energy input in the form of ATP and NADPH. Next question, ATP calculation. Calculate the total ATP produced from the complex oxidation of 1 mole of palmitic acid, or C16-0. The complex oxidation of palmitic acid produces approximately 106 ATP. This includes ATP generated from beta oxidation, or NADH and FADH2, and from the oxidation of acetyl-CoA in the Krebs cycle. However, 2 ATP are used during the activation of fatty acids, so the net yield remains about 106 ATP. The next question about triacylglycerol metabolism. Explain how triacylglycerol is synthesized, broken down, or lipolysis, and its role in energy storage. Triacyl- Triacylglycerol is synthesized from glycerol and fatty acid through esterification in the cytoplasm. During lipolysis, triacylglycerol is broken down into glycerol and fatty acid by lipase enzyme. Triacylglycerol serves as the main form of energy storage in the body, providing a large energy reserve that can be mobilized when needed. Next question about cholesterol metabolism. Explain the process of cholesterol synthesis and the role of cholesterol in the body. Cholesterol synthesis occurs mainly in the liver and begins with acetyl-CoA. The key regulatory step is catalyzed by the enzyme HMG-CoA reductase, which converts HMG-CoA into mevalonate. Cholesterol plays important roles in maintaining cell membrane structure, and it serves as a precursor for steroid hormones, vitamin D, and bile acid. Next question about regulation of lipid metabolism. Analyze the role of insulin and glucagon hormones in regulating lipid metabolism. Insulin promotes lipid synthesis and storage by stimulating fatty acid synthesis and inhibiting lipolysis. In contrast, glucagon stimulates lipolysis and fatty acid oxidation by activating enzymes that break down stored lipids. These hormones work together to maintain energy balance depending on the nutritional state of the body. The last question about integration of lipid metabolism. Based on the case, explain the relationship between fatty acid metabolism, triacylglycerol metabolism, and cholesterol metabolism in conditions of obesity and metabolic disorders. In obesity and metabolic disorders, decreased fatty acid oxidation leads to the accumulation of fatty acids, which are converted into triacylglycerol and stored in tissues. At the same time, increased synthesis of lipids and cholesterol contributes to high LDL levels, and this energy-interconnected pathway results in fat accumulation, fatty liver, and impaired energy metabolism as observed in the case. Okay, that's all for my presentation. Thank you.